The goal here is to essentially take the concepts that we've been talking about and then integrate them into this slide. Okay, so let's start off with the basics here. So cytotoxic T cell activation, which we kind of just talked about, very good versus intracellular pathogens. Again, we have our kind of everyday nucleated cell here. It's usually going to be presenting self-antigens on MHC1. However, if this cell was infected with some kind of virus, for example, right, like we just talked about, if there was some virus around that infected this cell, okay, here's my virus infecting the cell, then that viral antigens might be presented on MHC1, CD8 positive T cells, again, binding to MHC1. These are going to be cytotoxic T cells. They'll recognize that this cell is infected by some intracellular pathogen, and they'll release perforin and granzyme B to destroy the cell. So perforin, just think of perforating the cell membrane, right? So these are essentially going to be molecules that are going to be cutting holes into this cell to essentially destroy it, to prevent this virus from hijacking more of the cell machinery and eventually leading to a terminal lytic cycle in the cell and then kind of reproducing accordingly. And so that's what the cytotoxic CD8 positive T cell is doing. It's trying to destroy the cell because it's infected, again, through this process that we just talked about. So I think we've hit on that a couple times. Now, the important thing to remember is that, again, the antigen that's presented was endogenous. In other words, it came from inside of the cell. It was a viral protein. Usually we expect self-antigens to be presented in MHC class 1, and normally our body should not be binding to those because we went through negative selection. However, think of one of the correlations of this, and this is kind of some next level stuff, but if you have a patient that has a transplant patient, for example, or you put a donor graft in a patient, so let's just say you put a graft in a patient, and now that patient has somebody else's cells, right? And if that person doesn't have, the same type of HLA receptors there, right, they're going to present different self-antigens. And so what's going to happen is if you put a graft in another patient and they have different MHC1 receptors or different variants of these, right, different HLA variants, and the person that received the graft, right, that person has T cells, right? They have CD8 positive T cells that could attack the grafted cells. So you can imagine, let's say that this cell, right, this cell came from the donor. Okay, and then this CD8 positive T cell, let's say that this was from the host. Okay, so this is the host or the recipient that received the graft. So what can happen is, again, this MHC receptor might be somewhat foreign, right? It might not have the same exact HLA compatibility. And so these host cells will recognize, hey, this is not a self-antigen, and they'll actually start to destroy these donor cells for that reason. Okay, and so this is something that you'll sometimes see. And then again, there's variants of this. We'll talk about all of this in transplant rejection, but essentially this is what's happening. If you don't have self-antigens on MHC1, those cells could get attacked. And that's why we immunosuppress patients after they get a kidney transplant or bone marrow transplant, because you don't want the immune system attacking these cells. But then that does open the door for a virus to potentially start to infect these cells without any regulation if you're immunosuppressed. But I'm going to get off the soapbox there and we'll go back to the next topic here, which is going to be the helper T cell activation. Now, one thing that's going to be a little tricky here, when you're talking about helper T cells, you can go two different routes. You can go a lot of different routes, actually. You can go T helper 1, T helper 2. Those are the big ones for step 1. But then, of course, there's regulatory T cells, there's T helper 17 cells, etc. But what I just told you earlier, okay, and again, we're just kind of adding layers to this, but what I just told you earlier is usually when you're talking about T helper cells, number one, this is CD4 positive, meaning that there's going to be MHC2 binding to CD4 positive T cell receptors, and this is usually going to be from an antigen presenting cell, right? An antigen presenting cell binding to a CD4 positive T cell, okay? So that's kind of what we've already talked about, and you can see it here. Here's the MHC2 receptor, here's the CD4 positive co-receptor, here's the T cell receptor, but there's also going to be a B7 CD28 binding, okay? And so essentially this is going to be a co-stimulatory signal. It's essentially going to help activate this T cell. Okay, and without that co-stimulatory signal, there's going to be a significant drop off in the ability for these T cells to get activated. And so this is just one of those things that you want to be familiar with. Now, B7 can also be referred to as CD80 or CD86. Okay, so just kind of keep that in mind. And a lot of people remember this as saying, look, 7 times 4 is 28, right? 7 is B7. 28 is what it's binding to, and the 4 is a CD4 positive T cell. Okay, so again, these are all kind of ways to, just remember this, because a lot of these are kind of confusing, and it might help to use some flashcards, go through some of the recall questions on here. But again, that's the general concept. So antigen presenting cell presents an antigen via MHC2, like we talked about, to a CD4 positive T cell, and there's a co-stimulatory signal that's B7 CD28, right? 7 times 4 is 28. Now, the thing that I told you earlier, I was kind of only giving you the half truth here. In general, when you stimulate helper T cells, there's usually going to be a very effective response versus extracellular pathogens, right? We talked about this. If it's an antigen presenting cell, usually like a macrophage, right? They're usually going to run into things like bacteria, okay? And they'll digest this bacteria, right? They're going to eat up this bacteria, and then they're going to put the bacteria in these phagolysosomes that will essentially get acidified. Once the invariant chain kind of pops off, they'll present these antigens on their MHC2 receptors to T cells. Okay, and again, these were extracellular pathogens that they're presenting, right? So this was an extracellular pathogen. And usually what will happen is in these situations, these antigen presenting cells will then release interleukin 4, like we talked about earlier. And the interleukin 4 will stimulate this T cell to become a T helper 2 cell. Okay, so again, this is going to be stimulated by interleukin 4 released from the antigen presenting cell. And then these T helper 2 cells, right, once this becomes a T helper 2 cell, that T helper 2 cell can then go on to release more interleukin 4, it can release interleukin 5, it can release interleukin 13. Okay, and we already kind of talked about some of the implications of this. These are all going to help stimulate eosinophils, basophils, mast cells. They're going to help cause class switching on B cells, which we'll talk about in the next video. Okay, so that's kind of the whole process. So when I think about helper T cells, this is really the process that I'm referring to when I'm thinking about helper T cells. Now, but we can't forget that there are also T helper 1 cells. You might be saying, well, how is that different from the T helper 2 response? Well, the T helper 1 response is pretty interesting. The T helper 1 response, let's just erase some of this here. So in the T helper 1 response, remember the difference is that we're going to be using interleukin 12 to stimulate this naive T cell to form a T helper 1 cell. Why would this antigen presenting cell want to release interleukin 12 instead of interleukin 4? Like, what would cause that? Well, let's just say, for example, that I'm dealing with an intracellular bacteria. Okay, so something a little different. Let's just say that I have an intracellular bacteria here. Okay, so not talking about a virus. What intracellular bacteria is very classic that we talk about very frequently? How about Mycobacteria tuberculosis? Okay, I'm just going to put MTB here for tuberculosis. Okay, remember, this is acid-fast intracellular bacteria, and a lot of times our body doesn't know how to destroy tuberculosis, right? And so this is going to be an intracellular pathogen. Okay, so it's intracellular in the antigen presenting cell, not necessarily affecting all of the cells systemically, but more classic for antigen presenting cells. But again, it's intracellular, right? The body digested this bacteria, but it doesn't really know what to do with it. It's having a hard time breaking it down. And so it's just going to kind of hold it here and present some of those antigens. And in the process of doing that, it's going to say, hey, we're dealing with something different here. It might release interleukin 12 instead to this T cell, causing this T cell to become a T helper 1 cell instead of a T helper 2 cell. Now, once we get a T helper 1 cell here, it might stimulate this macrophage and say, here, let me give you some extra juice. It'll give it some interferon gamma to activate this macrophage. Okay, and again, these macrophages can eventually go on to form epithelioid macrophages, giant cells, and the whole idea is we're essentially going to use these macrophages to kind of help digest and wall off this infection. And that's where you get your granuloma formation, okay, which is very classic for this T helper 1 response. And the key is that this all kind of happened because, again, this was an intracellular pathogens. So in the last video, when I talked about MHC complexes, I said, in general, if you're talking about CD4 positive cells, you're usually talking about extracellular pathogens that you're digesting. However, with the T helper 1 response, this is more so specific for intracellular pathogens. So I know that's kind of confusing, but just keep that in mind. And usually this is going to come up when we talk about things like granulomas or viruses that are potentially infecting antigen presenting cells. And these T cells will go on, like we said, to release interferon gamma to the macrophage to activate the macrophage, and we'll also get some interleukin 2 release and then TNF alpha. And remember, the other thing I want to say here is interleukin 2, when we talked about cytokines, you're going to get some interleukin 2 release pretty much with almost all of these different steps. Interleukin 2 is going to stimulate all immune cells, T cells, B cells, natural killer cells, etc. And the key thing to remember the high yield thing about interleukin 2 is it can also have autocrine action. It can actually kind of stimulate the same cell that released it. Okay, so we talked about that in the cytokine video. Finally, the last thing on here is going to be these regulatory T cells. And regulatory T cells, like I said, they're kind of going to help damper down the immune response. These are essentially going to come from CD4 positive T cells that had some binding to self-antigens, okay? And so these regulatory T cells are very cognizant of that. So they're protecting us from self-antigens, preventing autoimmunity. That's the key with regulatory T cells. So you can imagine if you had some defect in your regulatory T cells or in the process of negative selection, you might end up with an autoimmune phenomena, right? So you can see the CD4 positivity in these cells, which kind of tells you where they came from. But the key to look for if you're trying to distinguish these cells from others, CD25 positive, FOXP3 positive are very classic for regulatory T cells. And again, if we're bringing down the immune response, right, we're going to be releasing things like TGF-beta. Okay, this is kind of like the opposite to the interleukin 2s of the world, the interleukin 1, the interleukin 6, right? This is going to be the complete opposite response. Don't get TGF-beta confused with TNF-alpha, right? TNF-alpha, we're stimulating, upregulating immune response, TGF-beta, downregulating immune response. And again, these regulatory T cells will be inhibited by interleukin 6. That should make sense. Interleukin 6, pro-inflammatory, TGF-beta is going to be anti-inflammatory. Now, once these cells are stimulated, they can release more TGF-beta, right, to increase the anti-inflammatory response, but they'll also release interleukin 10. So the big two to remember for anti-inflammatory response is going to be TGF-beta, interleukin 10 to decrease the immune response. Kind of a lot of information here, but I think this is really pretty much all you need to really nail down T cells, even though it's such a big topic. And in the next series, we'll take this one step further with B cells to see how we end up developing antibodies.